Okay, so today we're talking about muscle tissue. We're going to talk about muscle tissue. There's some things that we're going to go over that are very basic, and then we're going to get down into the actual tissue of the muscle, and we're going to learn how muscles shorten. And when muscles shorten, that's called a muscle contraction. So first, let's look at the anatomy of a muscle. And so this is a picture. I also have a drawing that we're going to kind of go over and label, but right now I just want to show this color picture so you can see. So we look at this right here, and this is showing a muscle. This is a whole muscle. So all of this is a muscle. So a muscle would be like your biceps brachii or your triceps brachii. That's an entire muscle. And the muscle is made up of smaller components. So the smaller components of the muscle is what we're going to go over right now, okay? So if we look at this entire circle here, okay, that's the entire muscle. And inside that muscle, you have muscle cells. Now, the muscle cells are a little bit different than the other cells we've been looking at. The other cells we've been looking at are kind of round or rectangular shaped or square. These muscle cells are really long. And so your biceps muscle runs the length of your arm, right? So your one cell is going to be quite long, up to 30 centimeters. So we can have really long muscle cells. But the muscle cells are going to be bundled up inside the muscle. So we look at this whole muscle, and on the inside of the muscle, we see these bigger circles here. Those are bundles of muscle cells. And we call those bundles of muscle cells fascicles. So those are bundles of cells. And it's these bundles of cells that make up the entire muscle. Right? Makes up the whole muscle. So inside those bundles of cells then, so this is a telescoped out bundle right here. Inside that telescoped out bundle, that fascicle, we have an actual muscle cell. Now because that muscle cell is long, instead of calling it a muscle cell, we call it a muscle fiber. A muscle fiber. So fiber is the same thing in this case as a cell. It's just an elongated structure. Now we also have connective tissues that surround each one of these three things. So we have a connective tissue that surrounds the bulk of the muscle on the outside, right? So that surrounds the whole outside. It wraps that muscle up into a nice little neat package. And that muscle, that connective tissue, we call the epimysium. Epimysium. That surrounds the whole muscle. Then we have a connective tissue that surrounds the fascicle. So here's the fascicle, the bundle. So there is a connective tissue that surrounds that. So if we look on the inside, we can see that it's surrounding each of the fascicles inside that muscle. Right? And so that we call the perimysium. Perimysium. That surrounds each individual fascicle. Then we have the individual cell itself, and there's a connective tissue that surrounds that. So it's all on the outside of that. And so we can see it surrounding, we can see it on the inside of the fascicle surrounding each one of the cells inside the fascicle. And we call that the endomysium. So these are all connective tissues. Endomysium surrounds the cells, perimysium surrounds the fascicles, epimysium surrounds the entire muscle. And all of those connective tissues at the end of the muscle, when the muscle cell ends, which is, you know, all of the cells have to end, so they're going to end right there. Those connective tissues continue on, and the connective tissues become the tendon that will attach to the bone. So we've got the epimysium, then we have the perimysium, then we have the endomysium, and they all come together to form that tendon. That's what forms the tendon. So this is the picture that I drew you, right? So you would want to be able to label everything on there. This is the entire muscle. It's covered by epimysium. This is the fascicle, which is a bundle of muscle fibers. And it's covered by perimysium. And then this would be the muscle fiber, which is the cell. And this is covered by endomysium. And after the cell, the cells end here, the tendon is made up of all three of those: epimysium, perimysium, and endomysium. Okay? So that's the structure of our muscle. Now we have to take a much closer look, though, at the muscle fiber, because the muscle fiber is going to contain these myofilaments, and you've heard of these myofilaments before in Chapter 3. Those myofilaments are called actin and myosin, or thin and thick filaments. Okay? So we have to take a look at those and see what those are. So this is just a picture here showing the muscle cell. So this is we're talking here about skeletal muscle. So this is a skeletal muscle fiber. And there's a couple of things that we can note here. If we look at the cell itself, the muscle fiber, we see, first of all, that there are multiple nuclei. We see one there, and there, and there, and there, and there, and there, and there. Right? So we would say that skeletal muscle is multinucleated, because that one cell has many nuclei. So we say it's multinucleated. Also, when we look at that cell, we can see that it has a striped appearance. And so we call that striped appearance striations, or we'll say that that muscle cell is striated. And it is those myofilaments that we're going to talk about, the actin and myosin, that give it that striped appearance. Okay. The other thing that we take note of are these things on the outside of the cell, and that is called a myosatellite cell. And the myosatellite cell can help to do some repairs to that muscle cell if it gets damaged. Okay. So let's now take a look. There's some other organelles inside here, of course, besides the nucleus. So we have a lot of mitochondria. So the mitochondria can produce ATP so that skeletal muscle fiber can shorten using the ATP. And we're going to go over how it uses ATP. But there's a lot of mitochondria in there. Remember that mitochondria make ATP, all right? And now we're going to look inside the cell. Take a bigger picture here and look inside the cell. So this is just a bigger picture of inside the cell. And so there's some things that I want you to see in here. So this is a muscle fiber. We're at the tiniest level that we can get at, right? The tiniest level. And inside that muscle fiber are those myofilaments. So this thing here is a telescoped out myofilament. Okay. And we see on this picture, you can see some darker blue and some lighter red. Right? So each one of those are different filaments. The darker blue filaments, those are the thick filaments, and the thick filaments are made up of a protein that's called myosin. Myosin. And then the red filaments that are in there, those are our thin filaments. And those thin filaments are made up of a protein called actin. And it's those filaments that are going to actually cause the shortening. The thin and the thick filaments are going to slide across each other, and that's going to shorten the myofilament. That would shorten the entire muscle fiber, and then if you have more muscle fibers doing that, it would shorten the entire muscle. Okay? So we have to look into the process of how does that happen and what are the tiny steps that occur in order for that to happen. There's some other things on this picture that I want you to note. So each one of these things here, okay, those are each bundles. It's inside the muscle fiber, and these are the myofilaments, right? Each one of these things are containing those myofilaments. So when we look at each one of these, each one would have the same orientation. They would have the thick filaments, and then they would have the thin filaments. And we're going to look more closely at those a little in just a couple of more slides, okay? A couple more pictures. So each one of those has those myofilaments in them. Now the muscle fiber itself, so the muscle fiber or muscle cell, it has, like I said, it's multinucleated. There are a lot of nuclei in this cell, right? So there's going to be nuclei, pretty much scattered throughout this cell. So these nuclei have the ability to make more of those thick and thin filaments. It's just protein. So the nucleus, each nucleus can make more of those. So if you start exercising and you're lifting weights and you're building muscle mass, what you're doing, you're not making more muscle cells. You're just making more of these thick and thin filaments, and you're making your muscles bulge out because they have more filaments in them. Okay. All right, now we see another thing that's covering the top of all of these, and it's right here. So this thing it's like a lace sleeve over the top of the myofilaments, and it reminds us a little of our organelle that we called the endoplasmic reticulum. So since this is occurring in the muscle, we're going to just call that the sarcoplasmic reticulum. Okay. And the sarcoplasmic reticulum, that it comes to the end, the very end of the sarcoplasmic reticulum, right there. That's called the terminal cisternae. Terminal cisternae. So the terminal cisternae stores calcium. And we need calcium in order for that myofilament to shorten. So we need calcium in order for the whole muscle to shorten. So it stores calcium in there like 100 times, 200 times what you would normally find in the extracellular fluid. A lot of calcium gets stored in the terminal cisternae. And when the muscle gets a signal and the muscle's told it's time to contract, then that terminal cisternae will release the calcium into the sarcoplasmic reticulum. Okay? Once the calcium's been released into the sarcoplasmic reticulum, then it can get released from there and it'll start floating around right on top of those myofilaments. And it will bind to the myofilaments. So that we need that process to occur in order to have a muscle contraction. We have to have the terminal cisternae release the calcium so that it floods over the sarcoplasmic reticulum, and then it finally floods over all of the filaments in the muscle, right? Yes?
Is that what happens like when you give birth, or is that totally different?
So birth is more, oxytocin is a hormone that causes the muscle contraction, but you know, smooth muscle, which is the uterus, is a little different.
Okay.
It doesn't have the sarcomeres, it's not striated like this, but it's similar, but not quite. So these are skeletal muscles. That was a good question. These are skeletal muscles. So there's 3 types of muscles. We have the smooth muscles, we have cardiac muscle, and then we have skeletal muscle. Skeletal muscle are the muscles that make your body move, right? You can contract them, and they shorten, and they make bones move. They protect your abdominal ones, like protect your organs. Those are the skeletal muscles. Then we have smooth muscles, which we'll talk about a little bit when we get to the digestive system and to some of the other systems, but like the blood vessels. The smooth muscles surround passageways in the body, like the blood vessels, the digestive tract, the urinary tract. So they don't act quite the same way. They're not arranged the same way. And when they contract, they're going to help to either move things through the tube or they're going to help to churn things inside the tube. But we'll talk more about those later. This is a skeletal muscle. Skeletal muscle is attached to bone or structures within the skeletal system.
So when it releases the calcium, is that changing the membrane potential? Is that why we're able to?
So that's interesting, and that's a good question too. The calcium is not changing the membrane potential, but it has a very distinct purpose, which is what we're going to lead into and we're going to talk about as soon as I show you some of the other structures. Okay, the calcium, in a nutshell, the calcium is going to expose an active site so that the thin and the thick filament can interact together and cause the shortening of that filament. Okay, that's the short answer. So great questions. Let's keep going then. So we kind of have now the anatomy, right? We know what's going on in the actual muscle cell, muscle fiber, right? Okay. All right, so here I have just a picture of the skeletal muscle fiber. And it's only a short segment of it. It's not like the entire thing. It's just a really short segment of it. And in here, I'm showing those blue lines and those red lines. And remember again that those blue lines are the thick filaments, or myosin, and those red lines are the thin filaments, or actin. Okay? So let's talk about a couple of other structures that we see in here, where where the thin filaments start, they're going to start at this zigzagged line over here that we call the Z line. And we can see that we have a Z line on both sides. Okay, and that's where the thin filaments are going to start. And the thick filaments, they're going to start in the middle here at a stationary blue protein that we call the M line, right? So some of the thick filaments are going to extend this way, and some of them are going to extend this way off of that M line. So we've got, they're extending off both sides of that M line. Those are the myosin or the thick filaments. Okay? They're extending away. The thin filaments then, they're going to go in between those thick filaments. So we can see coming from the Z line, we have all of these thin filaments that are extending in between those thick filaments. Right? And then we see them from this Z line extending in between those filaments. Okay? This entire thing from Z line to Z line we call a sarcomere. A sarcomere. It's the functional unit of that muscle fiber. It's the one that's going to shorten and help to cause that entire muscle to shorten. This is just showing one sarcomere in this very short piece of the skeletal muscle fiber, but actually a skeletal muscle fiber, depending on its length, it's going to have many, many, many of these. There'll be another sarcomere extending off of this side, right? And there will be another sarcomere extending off of this side. Whoops. Okay. There's another M line. And we can have, there can be hundreds of these sarcomeres on one muscle cell, right? So they're adjacent to each other all the way down that cell. So what's going to happen? Let me get rid of all these. What's going to happen when this sarcomere shortens is that these actin molecules, the thin ones, the red ones, they're going to move closer to the M line. They all move closer to the M line. The thick filaments do not move. The thin filaments move closer to the M line. So what's going to happen to the sarcomere when those thin filaments move towards the middle? The sarcomere is going to shorten. And if you have 100 sarcomeres doing the same thing, then all of them shorten and that entire muscle cell is going to shorten. And if you have all the muscle cells shortening like that, then the whole muscle is going to shorten. That's a contraction. That's what a contraction is. Okay. So this is showing just a real rough sketch here of the sarcomere without it being in the muscle itself. So again, we have those thick filaments. And the thick filaments have a head on them. And so I've drawn in little heads that are attached to the myosin. And if those little heads can grab a hold of the actin molecule and then pivot and pull them towards the center, then we get a shortening of that sarcomere. So that's what we have to look at next. We have to look at the microscopic picture of the thin filaments and the thick filaments. We have to see what they look like, and then how do they actually grab on and pull those thin filaments closer to the center. Okay, so you see all of those myosin molecules have heads on them, and all of those heads can attach to those thin filaments and then pivot and pull them closer to the M line, right, on both sides. They have to be pivoted in a way that when they attach and then they spring, they can spring that thin filament towards the center. Okay? So is this, it's making sense, right, so far? Kind of? All right, so let's look at the actual thin filament. This is the thin filament. So this here is the actin molecule. This is actin, otherwise known as the thin filament. So we have to look at the anatomy. This is a protein inside, it's one of the myofilaments inside that muscle cell, right? It's the red one, if you remember, the thin red one. Okay, so each one of these black circles here, each one of these circles, those are the actual actin proteins. They're globular proteins, so they're in this circle. They're just a circular protein. And on each one of those actin molecules, we have this green area here that I'm showing. That's an active site. So the active site is on the actin molecule. That's where the myosin head is going to bind. The myosin head is going to bind to that, right? But we have a problem. At rest, when the muscle is just resting and has not gotten the signal, hasn't received calcium, doesn't know it's supposed to contract, that active site is covered up by this blue band here. And that blue band we call tropomyosin. 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 So it covers it. It covers the active sites. It's a pretty long rope, and so it can get kind of flimsy, so it has to be held down by something. So there's another protein that holds it down, and those proteins are here, and we call those proteins troponin. Troponin. Okay. So for a quick note here, and flipping over to cardiac muscle, when cardiac muscle starts to break down, when a person's having a heart attack, all the filaments, the cells start to break down, the filaments are going to break down, and troponin is going to get released into the blood. So it's one of the first things that they take a blood sample of and look for to determine if they're having a heart attack. Troponin levels, right? So they'll take blood and look for troponin levels. All right, but this is skeletal muscle, so let's go back to skeletal muscle. All right, so these troponins hold down the tropomyosin, and they cover up those active. They keep the tropomyosin covering those active sites. Now, something can bind to troponin, which will change the shape and move both troponin and tropomyosin away from the active site. And what do you think that is?
Calcium.
There you go. So calcium can bind. Do a different color. Calcium can bind to the troponin. And when calcium binds to troponin, it changes the shape, changes the molecule, and the tropomyosin rolls away from the active sites. And now we have an exposed active site. Well, that's where the myosin wants to bind. And the myosin can't bind unless that active site is exposed. So that's exactly what we need to start this process, to start the sarcomere shortening. We need calcium to bind to troponin, to move tropomyosin away from the active site, to expose the active site so the myosin head can bind. So the questions on the exam are going to be, what does troponin do? What does tropomyosin do? What can expose the active site? What ion will expose the active site? It's calcium. So those are going to be some of the questions that you'll have on the lecture exam. So when you're studying, you want to be able to just take this picture and be able to explain to yourself, how does that active site get exposed? What are the functions of each of those little components that I just went over? And if you can do that, then you're ready to take the practice quiz on it, right? Okay, let's look at the myosin head then. Let's look at the myosin molecule. So the myosin molecule is also a protein. And so this is the M line. So the M line is a protein that's going to anchor that myosin molecule down so the myosin molecule does not move. It's the thin filaments, the actin, that move towards the center, but the myosin molecules stay stationary. They don't move. So it's this M line here that's going to keep them stationary. Keeps them stationary, right? So with the myosin molecule, there's this long tail. Then there's a little spring. Then each myosin molecule has 2 heads. Okay? And so that spring then, the spring can cock the myosin heads. So if the heads are facing away, like this one is facing away, then we say that the myosin molecule is activated. That's activated myosin because the heads are cocked away from the M line. So on the other side over here, you see the heads here are cocked away from the M line. So those are also activated, right? Because they're cocked away. It's the little spring that has the ability to become cocked, and then it also has the ability to spring the other direction, right? These myosin molecules down here, those are myosin molecules that have already been sprung, right? So they've already, what we call, pivoted. They've already pivoted. They were once activated, then they sprung, and now they've pivoted already. So now we need something to activate them again. We need something to recock that spring in there. All right, we need a break. All right, so let's continue on, and I want to show you how the actin and myosin molecules, how they interact with each other. And then we're going to finish up with that, and then we'll go into the rest of the physiology on Monday when we come back, okay? So let's look at, I got a clean sheet here. I'm just going to draw out one strand of those actin molecules so that we can see them. We're going to go over that. So this is just one strand. So the thin filaments are going to have 2 of these strands. Those are all the actin proteins. And then we have the active sites over the top, right? So those are the active sites. Then we have that tropomyosin covering all those active sites. And then we have the troponin holding it down, right? So this is a cell at rest. This is a muscle cell at rest. It's not even thinking about contracting because those active sites are covered up. But as soon as we get calcium being released, and where's calcium released from?
The terminal cisternae.
So it's stored in the terminal cisternae, it gets released from the terminal cisternae and goes into the sarcoplasmic reticulum, and from there it diffuses out and it floods over the top of the thin and thick filaments. Okay? So that's where we get the calcium from. So when when the calcium binds, then we end up with an actin molecule that's going to look more like this. So we've got the active sites, right? Now those active sites are exposed, and the tropomyosin is going to be down on this end with the troponin still attached to the tropomyosin, but now we can see that all of those active sites, they're all exposed. They're not covered up anymore, right? So that's going to be the shortening of the sarcomere. That's going to be the very first step that has to happen. So we have to have calcium, right? So now let's look at what we call the sliding filament theory. It's also known as the contraction cycle. And we're going to look at how the myosin molecule interacts with that actin molecule. So I'm just going to erase this because it's in my way. Okay, now let's say that the M line, we'll put the M line over here. There's our M line. And here's our myosin molecule out this way. And I'm just going to do one myosin molecule. I'm not going to do a number of them, which is what you would have. But this is one myosin molecule. And we start with this myosin molecule with the heads in a cocked position. So that means they're cocked, they're away from the M line. Now, ATP comes into play here. So I'm adding a layer of complexity first, all right? ATP is going to come into play. But first, as soon as those active sites are, as soon as they're exposed, the heads can bind right to them. So that's what's going to happen. The head will bind right to it as soon as that active site is exposed. There's like this chemical attraction and boom, they just bind right together. Okay, so that's a passive process that happens, right? Now the myosin head though, this myosin head is already in the activated position. And in order to be in the activated position, we had to break apart an ATP molecule. Okay? We had to break an ATP molecule apart. So if you remember, so if this were a head here, so this is a myosin head here, or this is the myosin molecule. And this is where the heads are already sprung, right? They already sprung. Well, that happened, after that happened, an ATP molecule is already bound there. The ATP molecule has to split. And it splits into an ADP and a phosphate. And the ADP and the phosphate are still bound to the myosin head, but that splitting of the ATP molecule allows this neck in here that has a spring, it allows it to recock, because we use the energy of breaking that bond to recock that myosin head. So now we're in this position here where the myosin head is in the cocked activated position, and it still has an ADP attached to it, and it still has a phosphate group attached to it. Okay? Now the ADP is going to get released, and the phosphate group is going to get released, and two things are going to happen. When the phosphate group gets released, it makes this bond here tighter. So it just makes it tighter. When the ADP gets released and that bond is broken, it's going to cause this myosin head that's attached, it's going to cause it to pivot, and then it will be in this position. So when it does that, it's carrying this actin molecule along with it. Right? When it does that, when it carries it along with it, it'll then be in this position. It has to be reactivated, so another ATP will bind to it. The ATP will split, and it'll go back in this position. Now it's going to grab onto an active site even further down the line, and it's going to pivot. It's going to continually do that. I'm going to show you a video here in just a second, an animation, so that you can see that. But the myosin head, so you have your actin molecule, and the myosin head attaches to it and pulls it, and then lets go. And then it attaches to it, pulls it, and then lets go. And recox attaches and pulls it and lets go. All right, so I'm going to do a demonstration, and then I'm going to have you guys watch a little video, and then that's going to be it for today with this, and then we'll start up on Monday doing the rest of it.